unfortunately, it was our first time doing a seminar, but we intend to make this a new tradition to invite former members back to talk about what they have done after their bachelor's degree. So I'd first like to thank the executive board for helping us get set up. I am the president, and my name is Tatum Pedrick, and can the rest of the executive board introduce yourselves to the audience?
Hi, my name is Kayla. I'm vice president of the Literature Society.
Hello, my name is Nathan, and I'm the secretary.
Hello, my name is Laura. I'm the Vice President of Social Media.
Hi, my name is Martha, and I'm the Vice President of Media.
Great. And today we are honored to have Akshay here. He was a former SLS president, and he is going to discuss today his journey through potentially med school and then research. He pursued biology and chemistry here at Nova, and he has moved on to obtain a master's degree and is now in his doctoral research on pesticides and how they affect synaptic transmission. So, Akshay, take it away.
Tatum, thank you. Tatum, thank you for that introduction, and Scientific Literature Society, thank you so much for inviting me. Dr. Shambag, of course, and Rashad, thank you very much for the invitation. It's a pleasure. So I've really divided my talk into two sections. I'm going to talk first about my research for some of the professors and so on that are in the room that might be interested, and for you students. And then we're going to shift gears a little bit about halfway through, and then I'll talk to you a little bit about my academic journey so far and how that has changed and evolved over time. My project when I entered Dr. Dawson Scully's lab was going to be a medicinal chemistry project and drug discovery project. I was very excited to do two things that I really loved, which was, going off of the medicinal chemistry research I did with Dr. Shambag and learning some new techniques with Dr. Dawson Scully. But in my first semester, I got sent this article, someone sent it to me, about Roundup. And I said it'd be very interesting if I looked at how Roundup would affect synaptic transmission in one of my classes, which was neurophysiology, which I took my first semester as a graduate student. And just offhandedly, Dr. Dawson Scully looked at me and he said, just check the literature. Someone had to have done it. There has to be data on it. And to my shock, there was surprisingly little published on this topic. So all this time later, fast forward 3 years, and this is my project. I study how Roundup affects synaptic transmission in two of our models. I'm going to focus on just one today, our C. elegans nematodes. And to give you just a bit of background on Roundup, if you're not familiar, starting in 1980, glyphosate was found to be this really great herbicide because it targeted a plant-specific amino acid synthesis pathway. Because that's not shared in animals, it was proposed at that time to pose no threat to animals or humans or anything. And fast forward a little while, and data starts to come out that glyphosate might not be this, inert, non-concerning thing. There might be some more nuance to it, especially when associated with Roundup. And a key distinction is when I talk about Roundup versus glyphosate. Glyphosate is the active ingredient. It is one chemical by itself. And it's doing all of the heavy lifting when it comes to herbicidal effect. But Roundup is glyphosate plus a slew of other chemicals, which is collectively known as the adjuvant components. These are stickers, buffers, anything else that would help Roundup function better. For instance, glyphosate might do a good job at killing weeds, but if the rain falls like it does here in South Florida, it might get washed off really quick. So you have surfactants in the adjuvant component to help the herbicide stick to the leaves of the weed so it doesn't get washed off very easily. When I was combing through the literature, this was the closest thing that I could find similar to anything I was interested in. It was a paper from 2012, which looked at C. elegans nematodes, which is the model I was working in, and they used two transgenic models. So these mutant models had GFP, green fluorescent protein, labeled dopamine neurons and GABA neurons. And they exposed these C. elegans to various concentrations of a glyphosate-based herbicide. It wasn't Roundup, but it was Roundup Light. Okay, it's called Touchdown. And what they found was, by exposing to different percentages of glyphosate, that they found neurotoxic effects when exposed to Touchdown. For dopaminergic neurons, they found that critical threshold to be 10% glyphosate, to be neurotoxic to dopaminergic neurons. And for GABAergic neurons, around 7% glyphosate. Reflecting on this project and taking it into context of what I wanted to do, I established my objectives and breaking down where the gaps in the literature was. So Dr. Dawson Scully had a novel seizure assay that he developed for C. elegans nematodes. It's a very robust screening model. So it's an invertebrate model, so there's a lot of nice screening things that you're allowed to do with that. And also, these C. elegans are very responsive to anti-epileptic drugs. So anytime Dr. Dawson Scully screened FDA-approved anti-epileptic drugs in this assay, they work. They recover the seizure behavior just like they would once it's a shared physiology between humans and C. elegans. So taking this model, I was going to expose the C. elegans to Roundup and expose them to dilute concentrations. That was the first thing that hit me coming from, again, heavy chemistry point of view. I said 7%, 10%? What's recommended on the label for humans to use recreationally in your garden is 2%. So you're talking about a microscopic nematode that's exposed to 3 times, 5 times as much as anything that you would ever come into contact with. Doesn't seem particularly physiological relevant to me. So I was going to take a dilute concentration and investigate my project. I was also going to compare Roundup formulations from the US and the UK and compare Roundup versus glyphosate. What does the active ingredient do on its own? And then what does the cocktail of chemicals do in that context? Also, there's legislation happening all the time about Roundup. You might have seen some of it. But particularly at the time when I started the project, the UK just placed a ban on a surfactant component that's usually found in Roundup called POEA, polyethoxylated taloamine. Did that surfactant have a significant effect on the seizure behavior? I wanted to find out. And of course, this is the big take-home of this assay. So I wanted to really use an anti-epileptic drug in conjunction with the glyphosate or Roundup components to see if there was any dualistic effects here, if there's any dance between these two components. A click seems so powerful until it isn't. There you go. All right. One of the first things that we take away from this assay is something called time to recovery. What does time to recovery mean? We take these little C. elegans, we put them in a tube. That tube is filled with either M9, anytime you see M9, that's normal saline. Nothing funny going on there, that's where they should be, that's how they normally behave, that's what they're usually accustomed to. Glyphosate, active ingredient only. US Roundup, same formulas all the way around, nothing crazy going on with US formulations of Roundup. The UK Roundup formulations, which I was able to accidentally get my hands on too. Incidentally, I got accidentally sent a bottle from 2015, which just so happened to be before they placed the ban on POEA, and then one in 2018 after. So I was able to compare those two together. And anytime you see NA-val, that is sodium valproate. That's our GABA agonist anti-epileptic drug. And what this assay does, time to recovery. C. elegans are swimming like this in the tube, and we shock them. An actual electroshock, 47 volts for 3 seconds. And their beautiful sinusoidal movement is disrupted, and the time that it takes for them to return to a normal sinusoidal movement, that is what we call time to recovery. These are our data, X-axis, all of our conditions, Y-axis, time to recovery in seconds. M9, of course, saline, about 35 seconds average time to recovery. Glyphosate, we saw a significant jump, well over 55 seconds time to recovery. The two US formulations of Roundup, again, a similar bump up to that of glyphosate, but in more the area of 60 to 70 seconds time to recovery. The two UK formulations, remember, this one 2015 is before the ban on POEA, the 2018 is after the ban on POEA. Again, didn't see anything funny there, but still, that jump, similar to glyphosate. And we took this formulation of Roundup, this 2019, the most relevant to us being here in the US, co-administered sodium valproate GABA agonist, FDA approved, and we returned the time to recovery back to the normal saline condition. And of course, this is our positive control with sodium valproate by itself. So that was a really significant discovery that we were able to return these extreme increases in seizure behavior back to normal conditions using that anti-epileptic drug. As is the case in most scientific projects, has been in the case of all of mine so far, you get that really unexpected thing that happens. So as I'm going through all of these videos, because we video record all of our sessions, as I'm going through, I'm seeing time to recovery seems to be an important, statistic. But after 5 minutes, which is the length of our videos, I noticed there were a significant amount of worms that just did not recover at all. And Dr. Dawson Scully and I met and said, we surely can't make them all 5 minutes because they don't recover. So we created a brand new category, a brand new bin for all of these, and it's our percent of non-recovery. So higher is worse on this graph, and x-axis has all of our conditions again to remind you what those are. In the M9 condition, it's about not even 5%. Glyphosate, 15% of worms don't recover. The Roundup formulations is where this gets extreme. US 2019, a third of our worms didn't recover from their seizures. So again, pretty significant results in not just the time to recovery, but in the percent of recovery. Less of our worms are recovering overall. But again, sodium valproate comes into the mix. You mix this and sodium valproate, and you get all of the worms recovering back again. So sodium valproate not only seems to have a significant effect on our time to recovery, but our percent recovery as well. This is a double check, all right? Between the UK formulations, it's very hard to precisely say, yes, this chemical is in this formulation of Roundup, and this chemical is in this formulation of Roundup. So what was the proof that POEA and that family of surfactants wasn't going to have an effect on this seizure behavior? First step of trickiness is that polyethoxylated taloamines are a family of compounds. It's not just one singular compound. So I used a representative compound, this POE15 taloamine, which was previously done as this control in other studies of Roundup. And at 0.065, that is what should normally be in 0.1 millimolar of Roundup, which is what we have been using so far in our study. So we did 10 times more dilute and 10 times more concentrated on either side. And this particular taloamine did not seem to have a significant effect on our seizure behavior whatsoever. On the flip side of this now, looking away from that adjuvant component and looking at glyphosate, that active ingredient, we tested all the time at 0.1 millimolar. But could we take this even lower and still find significant effects? We did this dosage curve starting at M9, 0, so this is no glyphosate at all, time to recovery on the y-axis, and at 0.001, no significance, at 0.01, nothing, and then at 0.1. This seems to be a critical threshold for understanding the results that we were seeing in our earlier bar graph. And not just the time to recovery. Even in this dosage curve experiment, we found that this percent of non-recovery stays well below 5% until you reach 0.1. 0.1 millimolar, then you jump up to this 15% in that area, which we reiterate what we found in the bar graph. The big question that all of the reviewers are asking me as I'm trying to publish this, what is the mechanism? What might be involved here? And the first step in answering this was sodium valproate. Sodium valproate has a mechanism of action. Sodium valproate targets this succinic semialdehyde dehydrogenase. This is how GABA is usually metabolized in the synaptic cleft. GABA transferase, you generate succinic semialdehyde, then the dehydrogenase creates succinate. Sodium valproate inhibits this dehydrogenase enzyme, which leads to an increase in succinic semialdehyde, which then inhibits GABA transferase, which then increases GABA at the synaptic cleft. So it's a roundabout way of doing it, but we get there. Just a couple hop, skips, and a jump. So whatever the mechanism is, increasing GABA neurotransmission seems to have a very significant effect on the seizure behavior that we are seeing. So now, my project is currently focusing on addressing my reviewer comments by going down this rabbit hole and looking at 2 mutant strains of C. elegans. One has depleted GABA neurotransmitter, so it's a GABA depleted mutant, and it targets this glutamic acid decarboxylase enzyme. And the UNC49, uncoordinated, so they abbreviate it to UNC. UNC49 has depleted GABA A receptor subunits. So by using these mutants, the hope is that for UNC25 or UNC49, if we see a baseline of X time to recovery, if Roundup and glyphosate are still pushing that time to recovery up, probably not by either of these mechanisms. But if it stays the same, GABA neurotransmission or the GABA A receptor is now implicated in the direct mechanism of glyphosate and Roundup affecting neurotransmission. PTZ is down there at the bottom. That's pentylene tetraazole. So I'm not using just a one-pronged approach using mutants, but 2, pharmacological. That's a GABA A antagonist. So it blocks GABA A receptors in normal C. elegans. So I'm using that as well to double check some of the results that we might come across. The key takeaways of this is that Roundup and glyphosate are significantly affecting the seizure behavior and non-recovery phenotype in our C. elegans. And this is tested at 0.002% glyphosate. So keep your 7 and 10% because this is created in my paper. I'm discussing this a little bit, this nuance. We often think of neurotoxic. Something doesn't have to be destroying neuronal tracts to have an effect. And I think that's what the data here is at least suggesting. That at a much lower level of glyphosate, you're able to generate some very, very nuanced effects. Treatment with sodium valproate alleviated both the seizure behavior and non-recovery phenotype. And my goal now is to work on mechanism. That's what I'll spend probably the next 3 years of my PhD looking into. So I'll pause here, take any questions you might have about the research project before we shift gears and go into the more academic career portion of this talk. Yes?
So I was wondering, like in the graph, I think you showed worms not recovered, right? Why do you think there's an increase for the 2018 version of the UK one after eliminating?
That's a really good question. And I think that highlights that, also I should say, this is the true data, so percent recovery, I find this the most meaningful. We're now working on transforming this data so I can actually run statistical tests and determine significance on these data. So after I do that, I'll be determining what's significant, what's not, because here, it might look significant, but I really couldn't tell you without running the stats whether that's a significant jump or not.
That was actually my second question, because, so normally you would, the statistics, like to find this, if it's significant or not, you would do it after?
Yeah, so without getting too statsy, because stats is not my area of great expertise, but you to run something like an ANOVA, you need variance. If it's just a percentage, there's no variance there. So just two numbers differing, it's not the right statistical test to run, right? There is no variance. So that's why this data, there's transforming data, which I'm working with a statistics professor now to understand how that process works. I'll actually be able to run some stats on it.
Got it.
Good question. Yes, Tatum.
So I see in this graph as well, USA 19 and sodium valve, they produce maybe even more of an effect for recovery than either the control M9 or just NA valve by itself. Do you know anything about that? Like you see how all worms recover with, for some reason, with pesticide and the anti-epileptic, but with just the anti-epileptic, they don't all recover.
Right. Now, could that be because this is just increasing GABA for normal worms, right? And that's going to lead to a certain percentage of non-recovery? That could be the case. But I'm not sure if 1, 2, 3 are different to 1, 2, 3, 4, 5, really until I run the stats.
Okay.
So I wouldn't be able to say whether those are significantly different or not. But if it were, I think that's a really interesting question. That'd be a really interesting question. Any questions from the professors?
Well, glyphosate or Roundup was banned recently in the US. Yes?
No.
Not yet.
No.
But there was a whole case and issue with its connection to cancer.
Yes.
Alright, so do we know what the mechanism of action there, that there may be a connection there?
No, nothing, because the cancer that's most closely associated with it are lymphomas.
Okay.
So not neuroblastomas or anything
Yeah.
to that effect, but there have been studies, small studies, to find that, especially farmers, people who are constantly exposed
Using it, yeah.
and not developing, there's the cancer studies, which is, again, one side of the literature, but there's another side. There was a large one done in Kansas and one done in France, where they find significant effects in depression symptoms, medicated depression symptoms for people. And one of the correlates that keep coming up, not insecticide, not fungicide, herbicide. Herbicide keeps coming back again and again. So again, might speak to the more nuance of this might not be a conversation for Parkinson's and neurodegenerative disease, but maybe behavior and mental health area. So that's a number of steps away from C. elegans, but it's certainly in amongst the conversation though.
So I'm guessing multiple pathways or like multiple mechanisms or modes by which this could act.
Oh, when you have the amount of chemicals that I'm sure are in that adjuvant component,
Exactly.
it could be anything, right? It could be anything, and glyphosate's a small part of that story. But, and I should say too, I'm not being purposely vague about what's in the adjuvant component. That's a trade secret from the company that owns Roundup. So, like the Coca-Cola formula or Big Mac sauce, they're not disclosing what's in it. So probably only the EPA knows that.
So the closest mechanism for here, what you're trying to get at would be, closest correlation would be what on the human side in terms of how?
In terms of GABA neurotransmission, should it be depleting GABA, it would have implications, especially GABA A receptors and that sort of thing, in behavior, linked to anxiety and depression and that sort of thing, which is interesting because those were the literature that they were founded in that. So if at a cellular level, C. elegans don't get depressed. we're anxious. But if at a cellular level you
don't know.
explain, if you explain on a cellular level though, what might be going on, that's the advantage of using invertebrates. Their genetics are easiest thing to manipulate and to create new mutants. So you can really target and answer this question of mechanism pretty nicely. So that's what I'm working on.
But I have a worry that the media attention and associated with
Exactly.
lymphoma
Yeah,
is
That's why I asked.
not because it's associated, but they could not dissociate it.
exactly.
They could not say it doesn't. I
Right.
think that's what the jury award is based on.
Yeah.
Is it affecting? No. Can you prove that it doesn't affect?
Legal arguments are different than scientific arguments. I think that's what.
That's why the
Right.
media has become.
I think that's where we're at on that. That's for sure. The political science is different to the to the rest of the science.
We're living it.
And if he gets too much political, his car tires will be slashed.
Yes, the black Mercedes will pull up and I'll disappear. So yes. Anything else? Any other questions? If
We
not,
can move on to next
we can
section.
move on to the next half of this. So, my beginnings here at NSU started out like I think many. Came here, wanted to go to medical school. I was fortunate to be in the dual admit DO program, and my whole freshman year went through, did all my classes, and that's where I wanted to be. That's what I wanted to do. Sophomore year started, going through it, and then second half of sophomore year, I ended up taking Organic Chem 2 with Dr. Shampag, and the part of osteopathic medicine that was really attractive to me was its openness to alternative modes of medicine. And hearing Dr. Shampag talk about his research was taking plant-derived compounds and synthesizing novel antibiotic candidates. That was right up my avenue. Exactly why I wanted to, be a DO and do all of that. So I asked Dr. Shampag, can I join your lab and start doing some research? And he said, volunteer and we'll see. And lesson number one I learned in research, every time though, you have to earn your stripes. No one's ever gonna give it to you, right? You have to earn your place in being there, and often it means putting in time that people aren't necessarily noticing, or you're getting paid for, or credit for, but it's important. So for that semester, did research, and Dr. Shampag accepted me into his lab for the next semester to do it with him, and that was so gratifying, going through my junior year and going through that process of being in a first research project, presenting at my first symposium, the ACS South Florida chapter. It was here at HPD that year, and the undergraduate student symposium. Really gratifying. I enjoyed it. I said, man, medicinal chemistry research, I'm doing this for the rest of my life. This is great. And I spoke to Dr. Shampag not too long before the end of the semester, said, all right, Dr. Shampag, these are my plans for senior year. I think we can do this, this, and this for the project. And he said, do something else. What do you mean do something else? He says, no, go see other things. Don't get stuck here. See other things and explore. He says, you're too young to stick in one thing. And I said, all right, what do? I was taking neurobiology with Dr. Munoz, and I said, wow, I'd love to do neuroscience research. This is really interesting too. And so I approached him. We had a whole idea for projects and all of that. And the research materials were either, there was an issue with that, and all of a sudden that project's not taking off. And he very graciously worked with Dr. Jamie Tarter to get me involved in her lab. And she took me under her wing my senior year, and I did neurogenetics, behavioral genetics research with her. Got to see cellular techniques, RT-PCR and genotyping, and then human subjects research as well. More psychology side of it, behavior, which is also part of neuroscience. And I really enjoyed that. I said, wow, this is really cool. This is really gratifying. got to work with master's students that she had in her lab, and I thoroughly enjoyed myself. Going through the process of applying to med school, secondary apps, MCAT, the whole shebang. And I chosen my path, I chosen a program over on the West Coast. I said, right, I'm going here. They even give me scholarship money. This is great. That's what I'm gonna do the rest of my time. And this was April. I'll never forget it. I'm hanging out back here at Parker, talking with some of my friends after, I'm sure, plant drug analysis. And I'm standing out there, Dr. Naraine, walking out to her car, Friday afternoon. Her and I have been able to catch up for a while. I tell her what's going on, and she's like, I just can't see you not doing research. I said, well, Dr. Naraine, I'll be able to do research. They have research at the university, and I'll take part in it. And she's just, that look of just, she says, but it's not your idea. You'll always be working on someone else's idea. You like research too much. And at that point, there's that feeling of, she's right, but I'm trapped. Right? it's where am I going to go? It's April, right? What am I going to do? And, you know, I knew it. She was right, and immediately she said, saw a look on my face, and she said, there's an opportunity to do an internship. It'll be unpaid, but it's for the summer at MD Anderson. You never know what happens. Consider it. Let me know on Monday morning. Like, all right. It's time. And over that weekend, I had to reflect. I talked to my parents, who are, you know, as far as they know, I'm bound for my program and all that stuff. Expectations, right? Everyone's got expectations, not just your own. And I spoke to my parents, said, I really feel that pursuing research is the right decision for me. I'm going to take a year, try to do research full time, and I'm going to apply again and do MD PhD. So I'm going to get something more out of taking my year off, right? Reinvesting it. Parents liked that talk. That was good. And I spoke to Dr. Naraine and Dr. Raja, and they just so kindly got me into that program, into that internship. And I traveled to Houston Medical Center, and got to work at MD Anderson. My first day there, I didn't know who I was meeting, who my PI was. I knew Tweardy Lab. I looked up, saw Dr. Tweardy's lab page and everything. I said, wow, okay, this is cool, this is neat. And I got to meet Dr. Prema Robinson, who was my direct supervisor. I still keep in touch with her. She's a really amazing scientist over there. And, you know, people probably tell you, hey, you know, if you're going to talk to a professor, you're going into a lab, do your research, read their papers, you know who they are. And I did that for this lab. I just read all their lab stuff, not their administrative page. So I came into that program not knowing Dr. Tweardy was the head of internal medicine at MD Anderson until I went to his office, and we're in the fancy building, and the elevator's going up and up and up, and I'm like, whoa, okay. And over the course of that summer, I didn't just learn Western blot, cell culture techniques, but imagine the workload of someone like that. I got to see a fraction of it, but just imagine seeing patients, being an administrative head of a hospital, the most famous cancer hospital in the world. You're the head of internal medicine there, and you're running a research lab, and you have like 1 billion projects in there. And he makes it all work like clockwork. And it's something that in my toughest days of the PhD program, I remind myself, my workload's never gonna be that. I better just get my butt moving, okay? Because he makes it happen. So I gotta make it happen. And a really motivating experience. He was a really dynamic mentor, and so was Dr. Robinson. Very dynamic mentors. Always an open book, always willing to share with me and tell me, because they were open books, I got to learn about what it meant to do research at a private research facility. And the stress that goes into that, because everyone is simultaneously doing research, and everyone is writing grants, which happens at most research-heavy places. But here, to even be considered, they built a giant glass triangle research building in Houston. To even be considered for lab space in there, needed 2 R01 NIH grants. 2, to even be considered. That's the most prestigious NIH grant that you can get. The most amount of money. You need 2 to even think about being considered for space in there. It's very competitive. And although Dr. Robinson and everyone there was very kind and everything to me, I was not a direct colleague to them. I saw how they were to the other scientists. Not that they were mean, but there was definitely a, I'm holding back, right? Not the most collaborative workspace ever to be in. And I picked that up right away. And I said, I really love and appreciate my time here, but cancer research is not really lighting my world on fire. And 2, I don't work in a space like that, right? That's just not who I am as a person. And until you're in it, you don't realize that. I was like, okay, not for me. I am way too talkative. I'll spill the beans somewhere. So, while there, I looked into what are called post-bac programs. These are research. You do research full-time. There are 2 really great ones that I saw at that time. More do it now. Max Planck, where I'm currently at, has one. Mayo Clinic has one. And the NIH has one. I applied to the NIH program, and again, learning experience. No one's in med school thinking is, I send my application in, they're going to review my application, and then send me a decision. When you apply to a post-bac program, you send in your application, and nobody touches it. Nobody is ever going to see that application, because you have to email the mentors and the PIs directly. If they're not pulling your application from the pool, it's not getting touched. So you better be reaching out and not sending mass emails to a bunch of PIs all in the email head and sending one generic email, because they'll get very upset with that. Individual emails. I read such and such a paper. This is what I did. I think this is how my skills suit your lab. I'm really excited to have a further conversation with you. Didn't hear back. Then all of a sudden it was, I'm not looking for a research student, but your skills suit my colleague. Is it okay if I pass on your application? Of course, please do. If there's anything else I can give, please let me know. I didn't hear back for like 2 weeks. I was like, nope, another dead end. And I was plugging away. All of a sudden, an email. I was passed on your application. I'm really interested. Can we talk tomorrow? Of course. And that's how I got into the NIH program. And it was very rapid. From undergrad, at a week, moved to Houston, get started there. After Houston, had a week, moved to Baltimore, Maryland, and started my research there. People asked me if I took a gap year. I was like, I don't see the gap. Like, there's no gap in that year. And I started a 1-year contract there at the NIH. So I like to pause here for a second, though, before I explain the NIH side of things. When I moved to Baltimore, I made another realization. Your academic career is one part of what you are doing. Your career path, yes, that is one part of what you are doing. Your life and what life lessons you are learning, do not be passive in that. And I think I was a bit more passive than I wanted to be in undergrad. And it wasn't until I was driving into Baltimore that I realized how much I was fooling myself. Driving into Baltimore, I reached their rush hour, and Siri said, save 30 minutes by taking this other route. That sounds good. Take it. It was through West Baltimore, which, if you don't know, West Baltimore is the 2nd most dangerous place in the US, next to St. Louis, Missouri. It has the highest murder rate per capita. Yeah. Longest 15 minutes of my life was driving through there. 15 minutes I drove through city streets, townhomes on both sides, didn't see a soul, didn't see a car, didn't see a convenience store, a mini mart, a Walmart, gas station, nothing. Nothing. Houses there, didn't see a single person. The scariest feeling of my life. I didn't stop at no stop sign, stop light, nothing. I'm just going, I'm looking, and I'm driving. Because it's crazy. It's just to think of, my parents are there from the Caribbean. I've seen the worst places there. I have family live in Bronx, Harlem, Compton, nothing like that. Nothing like that. And I realized that, in undergrad, we talk about issues, SLS, we debate, certain topics, the conversation we have, sometimes the conversation and how that revolves around science. I realized how sometimes it's just soft language that we use to describe things that aren't soft. They're real, and it's tough. War on drugs. What does that even mean? Every day I went into the NIH, I had to walk by the NIDA, National Institute on Drug Abuse, their free clinic. You actually see drug addicts and so on, kids, teenagers, moms, grown men, everyone. War on drugs doesn't cut it. That language doesn't cut it. What about the socioeconomically disadvantaged occupy substandard living conditions? Poor people live in the ghetto. And if it makes you feel uncomfortable, it should. Because there wasn't anything comfortable about that drive. And again, going through this, I said, damn, I was fooling myself in undergrad. Thought I know about any of this without seeing it. And let me tell you, you're here in undergrad to learn these things, to have these experiences and, learn and grow. Make sure you're doing that. Not just your academics, but you as a person too. That you're an active participant in that. Because let me tell you, if you're active, you might learn it here in the classroom. I can guarantee you're not learning it on an online class. Guarantee. Ain't learning it there. So that was lesson number 1, moving to Baltimore. I then got to work with Dr. McPherson and Dr. Fishbein in an MRI project, which, beautiful. Love this project. Neuroanatomy, this is the vasculature of a rat brain. Not using any contrast. No contrast agents in this. And it's that clear, that good. This was a novel scan that my postdoc, Dr. McPherson, developed. Really amazing. You don't get this kind of resolution without using gadolinium or some contrast agent. And as well as learning all of these things, how did I give up something like the NIH and start wanting to move from that? They gave really good health insurance, they paid well. It was a nice setup. I was in a lab meeting we had a collaboration with another lab, and not Dr. Fishbein, not Dr. McPherson, let me be very clear. There was another person, a higher up, and they put up a graph of data that I collected. I know what that data is, so does my postdoc, Dr. McPherson. That data didn't look like that. That data did not look like that. And I thought I was dreaming something. I looked at my postdoc, she had the same look on her face. I said, right, I'm not imagining this. And it was data fabrication. Plain and simple, that's the way I would describe it. And saw my postdoc go through all the right channels to do the right thing, and she was told, right, we understand your concern, but you really should have talked this over with that person individually, not go through these routes. And within 8 months, she was out of the postdoc program. Doing the right thing ain't always guaranteeing you the best outcome. But I still keep in touch with my postdoc and remind her all the time. You made the right choice. She landed on her feet, thankfully, and That is a testament to who she is as a person. But she made the right choice. She made sure that it wouldn't fall back on her or any of the postbacks that were in the lab, not just me. She easily saved 3 of us from ever being involved in any of those conversations. My career hadn't even started yet, and that could have been immediately a blackball. I was being forced to sign authorship things and all sorts of things. No thanks. And that's what pushed me. At that point, I hadn't received a single interview yet for MD, PhD. Nothing. So I made a decision. Either MD or PhD. I cannot have both. And even with all this mess that I've told you about, I loved research. I was excited. Looking at this, I did this work 4 years ago. 4.5 years ago. It still excites me. Still like a kid in the candy store looking at it. I'm very proud of it. And what even means my postdoc. And I said, I can't give that up. I can't give that up. Love medicine. It's a great career path. This is what lights my life on fire, is doing research like this. Reached out to a number of neuroscience PhD programs. The PIs, because I learned from my time at the NIH, don't apply to the program, apply to the person. Got in touch with Dr. Dawson Scully at FAU, well, at the time FAU, but at FAU, and he immediately emailed me back. Great, let's Skype tomorrow. I want to hear it. By the time we ended that Skype conversation, he was scheduling my GRE because I hadn't taken it yet. I was like, I took the MCAT twice, does that count? He's like, no, GRE. So GRE, for your information, if you haven't done it, much easier to take than the MCAT. Also, you don't have, it's not like the Hunger Games in October where everyone's trying to get a seat. I got a seat, I signed up for like a week later to take it. Easiest thing ever. I joined, and seriously, I've never looked back. I've never, ever looked back. It has been a fantastic experience, very gratifying experience to work and be a TA for AMP and work in an AMP lab like I saw my professors do, and get to collaborate and be in an environment where I'm happy. Mentor undergraduate students all the time. One of them's here. And it was really fun. And yes, I get teased all the time, to answer your question. One of my, that happened to me. I do improv comedy, well, before COVID. I did it all the time here at Falcon Pub. My friend told me this in the middle of a sketch. It was a game called Questions Only, where we have to talk questions like, hey, aren't you such and such? And he's like, aren't you the guy who didn't get into med school? Huge laugh. And yeah, I get teased. It was funny as hell though. But it was, but I'm happy. Med school isn't happiness, right? Or it doesn't have to be. So if you're thinking of other things, it's definitely the shift that I made. And again, thoroughly enjoyed it. So I'm happy to take your questions. I'd like to thank a few people real quick. Dr. Naraine is here. Dr. Shambag, thank you very much for inviting me again. And to all the amazing students, undergraduate students who helped me work on my research project for the data. Thank you so much. And for everyone who's helped me on my journey, thank you. And thank you SLS for inviting me.
On behalf of SLS and everyone here, we'd like to thank you for this amazing presentation. And if it's all right with you, I'd like to open the floor up to any questions for the first presentation, if there's any leftover questions, and from this one.
Yeah, let's do it.
I have a quick one. Where are your parents on it now that you are not going to?
They are, they just got a grandchild, so they're not wearing it. But no, it's, no, all jokes aside though, yeah, my parents, they just wanted to make sure I had a next step in mind, and I was pursuing something. And, one side was, I'm going to FAU because they have this great relationship with Max Planck. which is the research program that I'm now part of, it was wow. Once they heard about what Max Planck was, that was really the selling point for them. And they said, you're obviously happy doing this. Go do that. And we don't have to pay tuition and you get paid? That's nice. So, yeah, crippling student debt is at least med school crippling student debt. It's not something I have to worry about, which is nice.
Yes.
Right after you got out of bachelor's, you went to do, like, where did you go first?
MD Anderson first.
And then, so you hop from like place to place, cause I know you did the really cool MRI thing. So like, cause I'm just a sophomore, but I wanna do that same stuff, and I wanna but I don't know where to start. So like after bachelor's, like getting involved in something like that, like what's your best advice for them?
They're called post-bac programs, post-baccalaureate programs. I highly recommend them if anyone's interested in getting involved in research. You have to do research full time. That's all you do. You don't take tests.
First do some undergrad research.
Oh yeah, do undergraduate research, right?
You gotta do that.
Yeah, otherwise
your foot wet.
you don't have much of an application if you don't do it here. But do post-bac research. That's a good next step after bachelor's, and then you see if you love it. I was doing it 8 hours a day, 5 days a week, and I said, "This is great. I love this."
Actually, let me just put a plug in here. Anybody who's interested in doing something and taking Akshay's route, you need to look into the biology internships because MD Anderson is one of our summer internships. And that's what I got Akshay into at that time. A little bit late, but.
A little bit late.
And applications are open now. They're all up on the website.
What are they called? The what internships?
Summer biology internships.
Okay.
Yeah.
What are the prereqs for it? The internships?
What are the prereqs?
Oh, common sense. Okay.
So you don't have to take specific classes or?
You have to take specific classes, yes,
Okay.
and you have to be, at most a junior, because you have to be returning the following year.
Yeah,
At most you can be a junior.
Okay.
You have to be returning.
You have to be returning the following year.
Oh, this is, okay, I see.
Yeah.
So you snuck him into.
Yeah, we stuck him in.
Yes.
Ah,
Yes.
so you were supposed to come back.
Yeah.
I see. So
I saw him sitting on the fence and wobbling, so we needed to get him in somewhere, so yeah, we got him in.
Yeah.
So if you are a junior or sophomore, it's the right time to apply.
Yes, absolutely.
Yes. This
And
is
we're
it.
taking applications now for next summer.
And how many do you place normally? Maybe two?
Yeah, we replaced 2, MD Anderson gives us 2 seats. And the research hospital in Austria is our other internship, they also take 2 seats.
Do we still have Johns Hopkins? No, we lost that.
No, we lost that, they didn't have the class.
Rashad, sorry, hand up.
Yeah.
Yeah, so that was an amazing presentation. I really liked it. I think one of my favorite parts was when you talked about the drive to Baltimore. And one of the reasons why I really liked it was because you talked about how you realized that you were lying or you were fooling yourself, right? And that's a very significant change in perspective. So I know like there was something that triggered you to think that way, but what was your thought process when you were like, wow, like, am I fooling myself? What was the thought process? Like, what did you ask yourself?
First it was, let me get out of here alive, right? Luckily there were some people holding tiki torches and doing funny stuff in Virginia, like not 3 days before. So one time in my life I've been thankful for melanin. But it's, I was like, I'll take whatever happens on airports. This got me through this. But no, but it was that. It was the fact that I'd never been put in a position where I was like, I'm taking gas and, prostitutes running up to me and that doesn't happen. Had never happened to me before. So when you start to have these life experiences, right? Also the post-bac programs, I say, are nice because you're not having the weekend then studying for tests. If you're doing a post-bac in a different location, you're having experiences too on the weekend. You're actually able to go out and have a bit of a life.
Oh, it's not 8 to 5. It's not just the 8 to 5. Any research job is not 8 to 5. Never consider that.
But depending on, for postbacks and that sort of thing. Yeah, not,
thinking.
yeah. So, and the postback programs, the ones that I've experienced, are very strict, actually very strict about that, right? Because we're there as trainees, we're not master's students, we're not PhD students. It's a different culture, which, as I said, it's a good way of saying, I'm doing this full time, can I do more, right? But that change in perspective that you're talking about is when you go through when I went through experiences like that, I said, right, this is making me think in a way I've never thought before, or nothing I'd ever experienced before. It's good, right? It makes you think critically about your own life. And that is the trigger. When you go through, whether they're traumatic or good or whatever, having that experience should start branching your mind out in different directions.
Got it, so actually experiencing that thing.
Yeah. Yeah.
Well, it looks like 1 p.m. classes are starting. We have 1 a.m. classes.
Yes.
That would not be good, but I would like to thank Akshay once again for this
Thank you.
fantastic
Thank you, guys.
presentation. I'd like to also thank SLS and the audience for coming in, being here for our first seminar, and hopefully there will be more upcoming in the future of this semester, next semester, and we'll keep you guys on the lookout if there's anything else. So thank you once again. Don't want you guys to be late to your 1 p.m.s. And just another round of applause.
Hi, Dr. Naraine. Thank you so much for coming.